don't know that.
I have
I think it took a long time.
I was part of the coach that I know that we went to. I didn't went for a while. The next one called out on Humboldt Scholarship. And then so we're going to go in there. Yeah. It was like a great opportunity. I feel like really nice, but definitely works.
Yeah, she said. Stop me.
Step one. I know.
And
What is it? You have an additional? You can skip this step.
Yeah, you shouldn't have to do any of that.
Ah, you've pressed a key.
Yep.
Okay, skip.
Yeah.
So it thinks that's a key.
So weird. Should I just do this and see if it works? Oh, it does.
Yep.
Yeah.
Yeah.
Okay. It's up to you. Do you want, what do you want to do? Yeah. Okay.
It's not quite 4, but I think we can go ahead and get started. Does anybody have any announcements they want to make before we get going? No? All right. Well, then it is my pleasure to introduce our speaker today, Dr. Amy Trowbridge. We got our undergrad at University of Illinois, Urbana-Champaign, her PhD at University of Colorado, working with Dean Bowers and did a postdoc at IU and then in Germany. And she does a broad diversity of a number of different things, right? But they're all kind of centered around how environmental changes influences plant chemical profiles and the consequences for insect populations. And with that, I'll give it to Amy.
Thank you. Okay. Well, thanks so much for being here and thanks for the invitation, John and Scott. I know you've done a ton of work and it's been so fun to meet with everybody. And yeah, it feels like there's just never enough time. So hopefully I'll get to see some of you at dinner and maybe even tomorrow and just anytime. Just come to Madison. Okay, so today I'm really excited actually. I've been telling some people today that I'm going to talk about some new data. So this isn't really like a very canned presentation. It's really new, like off the press, hot off the press data that I'm really excited to talk to you about. So yeah, the broad title is Assessing Controls Over Tree Chemical Defenses, Consequences for Forest Function in a Changing World. That's a really big ask. But I'm going to do my best and I'll tell you some of the ideas that we have in the lab. Okay, so let's start here, thinking about the fact that global warming plus these episodic droughts and heat waves are resulting in elevated levels of both chronic and acute stress in trees. And then what we tend to see is these epidemics of forest insects and diseases emerging and lots of tree mortality across large forested areas across the globe, right? And so what we know is that drought-induced tree mortality is this global phenomenon, right? This is a map actually from Henrik Hartmann's paper in 2018, where every single dot on this map represents a major tree mortality event. And you can see that every single continent on the planet is affected by this, with the exception of Antarctica, but if there are trees there, I would love to know. And so this is a global phenomenon. There are really important consequences for, ecosystem function and services, but we really don't have a good sense of some pressing questions that remain in terms of like which species might be most vulnerable to drought, right? Drought-induced mortality in a given region, which individuals within populations might be at high risk. And then we don't have good estimates for these quantitative physiological thresholds that could be really useful if we're trying to predict mortality under future climate scenarios. So because we have these outstanding questions, this has led to a lot of research just trying to understand mechanisms of drought-induced tree mortality. In other words, we still don't actually know how trees die, which may be disappointing for a lot of us here. We've posed a lot of different hypotheses, thinking about trees dying from hydraulic failure, carbon starvation, and biotic agents. And what we're finding is that they're all very much interrelated, and trees are probably dying from a combination of all three. However, most of the research in this area has really focused on the effect of drought on carbon and water economies. And because of that, the models tend to follow the data, and lots of models are actually only considering forest mortality in terms of just changes to tree physiology responses, mostly water and carbon, and rarely, if ever, on insect responses to tree stress physiology and chemistry, right? So I've yet to see a process-based model that does include insects in a very meaningful way. So what we did is over COVID, this was like a COVID project where Nate McDowell got a bunch of us together and said, why don't we come up with a new framework and update the 2008 one that everyone has come to love and hate. And so that's what we did. And so this is kind of our most current understanding of how we think trees die from drought stress. And so I'm not going to go through all of this, but what I want to point out is that all of these stages are basically different different points along a drought mortality continuum. And what I've highlighted here in red is that defense actually pops up at every single point along the sequence as a major carbon sink, right? So even though drought is constraining acquisition of carbon, we still see defenses kind of being considered a major sink. And then what I want to point out here, so I've highlighted this in red, meaning it's important, but then I don't know if you've seen this down in the corner. These are the biotic agents. And so this is where our framework has come in the last decade, which is just now adding it onto the framework, and then biotic agents question mark. So we have added a question mark, meaning we know it's important, but we still don't know where it really goes in this framework. And so not only are we kind of confused as to how to integrate this, but we don't have a good sense even of how defenses or secondary metabolites are changing with drought. And we don't have a good sense of how those shifts are actually affecting tree resistance to various biotic agents. So this is a huge black box in our drought mortality modeling and frameworks. So what we do know, so I've told you a bunch about what we don't know, but what we do know is that tree chemistry does matter for insect choice. And what we'd really like to know, though, is what do susceptible trees look like? What are the phenotypes or the chemotypes that may allow us to make some predictions about who may survive and who may die under drought conditions? So I'm going to give you a little bit of a primer just on secondary metabolism. So what trees produce, hundreds of thousands of multifunctional metabolites. Really great image from my colleague and friend Laura Richards. So every line here is a different species within one genus of piper. And all of the different peaks, this is NMR data, all the different peaks are actually representing this incredible intraspecific chemical diversity that just exists in nature. Just this is, like I said, one genus. And so you can imagine then we have all of these chemicals being made, lots and lots of diversity. So as scientists, what we like to do is take all of that diversity and complexity and then just put it into boxes. And so when you look in the historical plant-insect interactions literature, we used to just think of all of these compounds as three major classes. So we would consider them either primary metabolites, so like lipids and carbs and things that are used in growth and reproduction directly. Secondary metabolites, where we used to think of them as just strictly defense compounds, and then hormones, which are just molecules that are regulating all these processes, right? But what we've really come to learn recently, and this is a great paper about this, is that all of these secondary metabolites that they make can actually serve as primary metabolites at some point. They can also be signals or regulating molecules in and of themselves. So they have lots of different functions. And so by dissolving this kind of functional trichotomy, what that allows us to do is really take a much broader perspective in terms of the function of secondary metabolites, and then emphasizes the need for us to understand the controls over their production, especially in response to different environmental stresses. I'm going to argue now that if we want to know how drought or other environmental stresses affect chemical defenses, we have to go a step backwards in the biosynthetic pathways and first understand something about non-structural carbohydrate dynamics. And this is because, as you can see here, so this is constitutive secondary metabolites, and this is induced secondary metabolites. They are not only produced just from recently fixed carbon and photoassimilates, they're also produced from stored non-structural carbohydrates, which are usually starch or soluble sugars, right? And so when you have a tree that is under drought, you have this constraint on carbon acquisition. And so now the tree has to, quote unquote, kind of prioritize or make decisions about how to allocate limited carbon to multiple functions, including reproduction, growth, perhaps active storage, but also to these defenses. And so the relative investment then into these defense compounds have really important consequences for herbivores and pathogens, which then have important consequences for tree function and fitness, right? So that's my argument. I'll leave it there. So what I'm going to do today, this is kind of just a broad intro, but what I want to do today is talk about two different field studies that my lab has been involved in over the years. The first one is going to be focused more on kind of how drought is affecting the chemical phenotype and non-structural carbohydrates in pinyon pine in the Southwest, and thinking about how that might help us predict bark beetle attack and resiliency during drought. And then in the second part of the talk, I'm going to move us back up to Wisconsin and talk about aspen, a broadleaf species, and thinking about how a different environmental stressor, this is going to be spongy moth defoliation, is interacting with genetics and how that is affecting the chemical phenotype, and what is the role then of NSC in determining how those defenses are modulated. Okay, so let's start here with can drought-induced shifts in leaf level defenses portend bark beetle attack. And this is interesting because if you know anything about bark beetles, and it's in the title, they do not affect the leaves, they actually bore into the bark and affect the phloem. So why am I even talking about this? Well, let's start here and just give you a little bit of background about this very cool study system. This is a system I've been working in for my entire academic career. So I think a lot about pinyon pine, Pinus edulis. This is a dominant conifer species in the four corner regions of the Southwest, which you can see here. And the primary biotic agent that causes mortality in this system is this cutie pie here, Ips confusus, the pinyon engraver. He's got like a little furry helmet. And so the thing about pinyon engraver is that they eat within the inner bark and the outer sapwood of the tree, just like most other woodborne species and bark beetles. But what they really require in order for populations to accumulate is a high density of stress trees across the landscape. So they're kind of wimpy. The trees have to be very, very stressed in order for them to enter. So this has been really great because it allows us to look at some of these thresholds. And this is unlike mountain pine beetle where they can become eruptive and actually enter even healthy trees. Okay, so pinyon pine and Ips confusus. Okay, so a lot of the work that we've been doing has been motivated by what we observed in the American Southwest in the early 2000s. So this is a picture taken by Craig Allen, and as you can see here, this is a pinyon juniper woodland. And at the time, the area was experiencing one of the hottest droughts, one of the most severe droughts on record. And what you see is a lot of pinyon entering the red stage here. You see some still in the green stage, some losing needles, and you see a lot of living juniper, right? And so if you were to just look at this picture in 2002, you might say, oh, well, some of these trees actually don't look so bad. Like they may survive, right? And so then Craig went out and took a picture 2 years later, and this is what the landscape looks like. And so this landscape has been drastically altered by drought and bark beetle infestations. And you can see that actually the only living vegetation here are the junipers. All the pinyon are dead. And so what would have been useful for us is to actually look at all of these trees when they were still green and start to make predictions based on chemical phenotype as to which trees might have greater or less success in terms of surviving drought-induced bark beetle attack. In this case, it might not have been as useful because all of the trees are dead. But what we typically do is kind of come in post-mortem and try to like piece everything backwards. So that's kind of my argument is, if we could have some sense of susceptibility beforehand, then maybe we can make some management decisions or at least understand some of the ecological consequences for the landscapes. So I'm talking about seeing shifts in phytochemicals, right? And so when I'm talking about that, I'm thinking about using remote sensing tools like hyperspectral leaf reflectance data. And the hope that we could use that to maybe quantify at least mortality risk from bark beetles. And that's true even if the chemistry is not explaining mortality, right? So the leaf chemistry is not going to explain how a tree is dying from a bark beetle, but it could serve as an early warning sign to tell us when trees are kind of shifting in their physiological state to make them more susceptible to bark beetles. Okay, so I'm going to tell you a little bit about this field site. This has been a labor of love. This is a field site. It's beautiful. This is in kind of south central New Mexico, just an hour south of Albuquerque in the Savieta National Wildlife Refuge. And this is a spot where we really wanted to identify how secondary metabolites in these mature pinyon pines are responding to drought and how those shifts are then altering insect behavior, like host choice, and also success. But that's really hard to do, to think about how to manipulate all that. So what we did is just manipulated the trees, and then we allowed the bark beetles, we like hired them to come in and then tell us which trees would be most susceptible. And it worked out great because you would be surprised at how difficult it is to actually kill a tree in the field. And we succeeded. And pinyon is like really hard to kill. Trust me, we've done like 4 of these experiments. Okay, so we killed trees. So now we can say something about mortality, which is exciting. Okay, so this is our control. This is what the site looks like. These are trees that are just getting ambient levels of precipitation. And now we have added these very sophisticated plastic gutter systems. Thank you, NSF. And so what we did is we added these gutter systems so that they would divert about 45% of ambient precipitation away from these mature trees in the field. But we also have this very cool legacy 45. So this is a plot that existed in 2010, and the trees were sprayed during a bark beetle outbreak to prevent bark beetle induced mortality. So these trees actually survived. They were the few trees that did in that area and have been allowed to acclimate to this mild level of drought stress over the past decade. So this really offered a very cool opportunity to think about acclimation and what that means for defense trees. Okay, so then we got real crazy with it and we added the 75% treatment, right? And so now we're diverting even more water away. And then we even got more nuts and we said, how about we just give them like no water? So this is the 90%. And really we had to have 10% because we couldn't put plastic over the trees or else we would have just done a mass. So anyway, this is what our treatment looks like. And then this is what we see in terms of mortality. So these were established in 2020. What I'm showing you here are the number of dead trees in 2021. So those were only in the 90. So they had only been installed for a year and trees were already dying. And then 2022, we see a lot more mortality. But what I hope you're noticing here is that in this legacy 45 that have acclimated to mild drought, we actually see 0 mortality, which is really interesting. So I'm going to talk a lot about the legacy plot today. Okay, so I want to first talk about phenolics, because that's what we're going to measure in the leaves. And I told you that there's hundreds of thousands of compounds, but we don't have time for that. So we're going to just focus on one group, these phenolics. And the reason that I want to focus on them is because if we're going to use remote sensing, leaf level remote sensing, I think phenolics might be a good candidate for compounds that can maybe link to shifts in physiological processes, and we can actually measure them and we have pretty good models for estimating them in leaf tissues. So what are phenolics? So they're characterized by this aromatic benzene ring, as you can see one right here. And then they have functional groups attached, usually a carboxylic acid or a hydroxyl group. And then we can add even more functional groups, right? And modify them in lots of different ways, giving rise to tons of structural diversity that leads to equally impressive functional diversity. And so in terms of biological functions, phenolics have lots of different functions. You might think of them mostly as kind of being structural when we think about lignin, right? Or something like that. But it's may be surprising to some of you, they can be involved in growth and reproduction. We know that they play huge roles as antioxidants, right? So scavenging reactive oxygen species that are produced under stress, especially drought. And also, maybe this is well known to you, plant resistance, right? Serving as kind of defensive compounds, maybe antimicrobial or anti-herbivore. And so when I usually talk about conifer defenses, I don't talk about phenolics. I usually talk about terpenes. And if you know anything about me or have seen me give a talk, that's what I love to get up here and talk about and wax poetic on. But today I'm talking about phenolics. So I'm living in this alternate reality and you're coming along for the ride. And so what we know about phenolics and conifers is that phenolics are produced in these parenchyma cells, in the bark and in the wood and in the roots and in the leaves, right? And so they're common. There's a wide variety of them that are produced, but the production of phenolics across species and organs varies with drought. And so I want to show you some kind of interesting results out of Nadir's lab at University of Alberta. So what I'm showing you here is phloem of lodgepole pine, foliage of lodgepole, and then we have phloem of jack pine down here. And water deficit is on the left, and then ambient treatments on the right. And what you can see, here is that under the drought stress, we see a reduction in both the phloem of the two species, but we actually see, an increase in the foliar phenolics. And what I think is kind of interesting here is that, or just a thought exercise is like, should we be considering some of these relationships between phloem and foliage phenolics? And I think we can make an argument for that if changes in the phloem chemistry, in the phloem phenolics, actually translates to variation in susceptibility to certain bark beetle species. And in fact, that's what we see in the European spruce beetle, right? It's Typographus. And so we see that trees that are characterized as having higher survival or being more resistant tend to have higher phloem phenolic compounds. So is that true in North American species? We have no idea, right? So in most conifer bark beetle systems, we still don't know how and when drought-induced changes in leaf phenolics are related to either phenolics in the phloem or other secondary metabolites that might be effective against bark beetles, like terpenes. And we just don't really know a lot about how phenolics affect bark beetle success in lots of North American species. So to your surprise, maybe mountain pine beetle is not the only bark beetle species in North America, although the literature may make you think that. Okay, so some questions that we had in this study. Can shifts in leaf phenolics predict bark beetle-induced tree mortality? And then can we use general spectral models to predict those traits? So what I want to show you here in the corner is just to kind of remind you about the level of mortality at the site. And what I'm showing you here on the right are leaf phenolics in the trees that died, the trees that survived that were in the legacy that acclimated, and the trees that survived that were in the other treatment groups. And what you can see here is that phenolics were increased in trees that died versus those that acclimated and survived. And so one possibility for this would be that under severe drought, there's a lot of reactive oxygen species that have to be dealt with. And so creating phenolics can serve an antioxidant role and help mitigate cell wall peroxidation and cell damage, right? That's just an idea. Okay. What's interesting here now is that phenolics, so I just showed you that they're greater in trees that died, and they also tend to increase with increasing drought stress. So they are related to water potential. This is water potential here on the x-axis. And so as trees become more drought stressed, we see phenolics increasing. This line here is just showing you the relative change in relation to the concentrations in trees that survive, right? So this would be equal to trees that survived, but we see the trees that died produce more, and that's because most of them are dying at more negative water potentials, right? Okay, so trees that die have higher leaf phenolics, but what's interesting here is that they're actually less responsive to changes in water potential. So these are just kind of the slopes from this figure on the left. And what you see is that trees that die do not respond, do not produce leaf phenolics as quickly as trees that survive or these legacy trees. So even though by the time they die, they have a lot of phenolics in their leaves, it seems that they might not be producing them as quickly as drought is happening, therefore maybe leading to more cell damage if reactive oxygen species are accumulating in the cells. And so then we did some logistic regressions, and what we find is that, maybe not surprising now, as leaf phenolics increase, the probability that trees die during the experiment increased, right? So that leads us to believe that maybe phenolics could be a pretty good predictor of tree mortality. So that's great, right? So if we have these phenolics in this trait and we can measure it and we know it's a good predictor, can we actually sense it remotely? Can we measure it remotely and get a good and be able to predict risk, right? And so what we did for this is we did leaf level hyperspectral reflectance measurements. And just for those of you who don't think about remote sensing very much, which includes myself, what we're really measuring is just light being reflected at hundreds, maybe even thousands of wavelengths. And the reason that we care about what's reflected is because chemistry, the different compounds absorb at different wavelengths. And so you can learn about unique chemical features of the leaf by looking at what's actually being reflected, right? So we get these nice kind of traces, these spectral traces. And we can learn a lot. It's jam-packed full of data and information. And so, like I said, we use this then to assess unique leaf traits and their unique responses to environmental conditions. And so what we did was we took leaf level hyperspectral reflectance data on all of our target trees during May and June, which is before the monsoon, which is important. It's really dry then. And then we also took it in August after the monsoons came. So they get a little bit of relief. What I'm showing is all of the reflectance data at the site over the 2 years, just to highlight the variation. I mean, lots of stuff is happening, right? There's lots of chemical shifts. So we're only looking at a very small portion when we're just targeting the metabolites like phenolics. Okay, so then, actually, let me stop here. So what we did was we took all this reflectance data and we put it in a general traits model that was developed using lots of conifer species and broadleaf species across different biomes, with the thought that you could just put these data into the model and you will get traits back, right? Phenolics back. And that this would be applicable to lots of different tree species in lots of different places. Okay, so I was really excited about this. And they were like, you're gonna get this really great positive correlation. It's gonna have a really high R squared. No, that is not. Look at this hot mess. Okay, so basically what I'm showing you here are the modeled phenolics here, normalized, and the measured phenolics here on the y-axis. What we see from the model is that it can, and I put in parentheses, predict. I mean, look at that R squared. That's not anything to write home about. And that should be positive. Sorry, I was late. Okay, so what we see here is a positive correlation, right? We see modeled phenolics and measured phenolics. Yes, okay, it can predict them, but not very well in trees that survived. Now, if you look at the purple here, this is the legacy, right? They don't predict them at all. And then when we look at dead trees, we actually see the opposite, which is super interesting. So if you have ideas, please tell me. And we see this negative relationship. And what this really means is that these models can maybe help us predict phenolics in healthy trees that survive, but does poorly in trees that are suffering from severe stress or have these kind of maybe weird or unique features because of acclimation. So in summary, phenolics we know are multifunctional, not just as defenses, but antioxidants, and their role in bark beetle defense in pinyon and other systems in North America is really, we just don't know. Leaf phenolics seem to be a pretty good indicator of bark beetle-induced mortality risk in Pinus edulis. But obviously, thinking about the relationship between leaf phenolics with shifts in woody tissue chemistry and how that really is directly affecting bark beetles is kind of the logical next step. And we've already been doing that, looking at both phenolics, but also a suite of terpenoids. Using a generalized spectral model worked better for survivors, but is not robust for dying trees or trees that may be kind of these weirdos that have acclimated, right? And this is likely because a lot of these generalized models, actually almost all of them, are actually trained using leaves from top of canopy, sunlit, healthy vegetation, right? And so they might not be robust enough to actually pick up on unique spectral features that emerge when trees are stressed or almost dying. Okay, so still staying at the Sebieta, we also wanted to think about NSC, right? So I've been talking about phenolics, a little bit about terpenes. So do higher levels of NSC translate to greater resiliency during drought and bark beetle attack? Okay, so this is work, I just want to recognize Drew Peltier, who is a postdoc on this project, is now going to be a faculty member at UNLV. This is all of his work. And it is multiple lines of evidence that trees in that legacy plot that I've been talking about, that have acclimated, that they are very much carbon limited or carbon starved. And so I'm going to provide you with some evidence here. Okay, so what I'm showing you here on the left, right? So this is measured CO2 flux or respiration. And so this is bowl respiration, stem respiration. And this is the control here in blue. This is the severely drought stressed trees, but only for a year in the 90. And then these are the legacy trees that have acclimated. So you can see that the respiration rates in these legacy trees are about 30% lower than the control trees, right? So they're respiring less. Whereas the trees that are just experiencing this brand new but very intense drought are like freaking out and they're respiring a lot, right? So clearly acclimation happening here. If you look here to the right, now I want to talk about how much carbon is even there. So this is how much carbon they're losing. How much carbon do they even have? And so this is starch on the y-axis here, the legacy, the control in the 90. And what you can see here is that the legacy actually has like less than 50% of the starch reserves relative to the control as well as the 90. So what this says is that, first of all, 1 year of intense drought stress is not going to reduce the starch reserves in these trees very much, right? They're no different than the control. But if you're acclimating to mild stress over a decade, you're probably having to use a lot of reserves to maintain, right, under stressful conditions. And so your safety margin is much lower. So they're respiring less carbon, they're losing less carbon, but they also have less carbon to begin with. Okay, so not only did we want to know how much carbon was there, but we also wanted to know the age of that carbon. And so what Drew uses is a technique with C14 radiocarbon. And so we've taken advantage of a terrible event, which was all this nuclear bomb testing in the 1950s. And so the atmosphere became enriched with 14C. And so you can use that now, the exponential decay, to actually age the amount or age the carbon that is in these trees, so because they've been taking it up over time. And so what I'm showing you here is the total age of sapwood NSC on the y-axis, legacy control in 90. And you can see that it's only reduced after these trees have been drought stressed for over a decade, right? And so these carbon reserves are relatively young, again, suggesting that they've had to use a lot of old reserves just to maintain under very stressful conditions. And then again, the 90, just 1 year of drought is not really changing the age of the carbon or the amount of carbon that's there. Okay, and the third line of evidence that I'm going to provide here as to how incredibly carbon constrained these trees are is just kind of more qualitative. So these are these white rings. So this is November of 2019, then 18, 17, 16, 15, back to 2011. And so what you can see is this color shift right here. And what's happening is that these rings here are exhibiting what we call white rings, which happens usually in trees that are heavily defoliated. And that means that they're usually carbon constrained and cannot lignify the cell walls that are formed during late wood formation, right? So this is just kind of another qualitative way to see that, yes, they are carbon limited. So these trees in the legacy, I've just given you like a lot to think about, but to summarize, they've survived bark beetle attack, right? This drought over 13 years, these bark beetles come through, they don't attack the legacy trees, even though they are very much living on the edge. They have very little reserves in order to respond to a biotic challenge or a disturbance, right? And I've given you lots of evidence for this. We see reduced starch reserve, the maximum age, right, is also reduced. This is very young carbon. And I've shown you some white ring formation. And so clearly they've acclimated and continue to survive. Why? Somebody tell me why. First of all, one question we have is, they have such a small carbon safety margin, right? And so what does this mean for resiliency under future conditions? I think I said to someone today, I felt like if a bark beetle just breathed on these trees, they were just going to fall over. That's how little carbon they have. So What happens when a biotic agent or another disturbance does find them on the landscape? How long is this sustainable? Like, how long can they acclimate to these conditions before, even something that we would not consider to be a disturbance actually takes them out? And how are they avoiding bark beetle attack, right? It could be that bark beetles, because they feed on the phloem and need carbohydrates, these trees are just too skinny. They just don't have enough there for them to really feed on. Maybe they can perceive that. Maybe there's something going on with their volatiles that masks them in the landscape and they can't find them. There could be a million reasons. So these are kind of some new questions that we're thinking about with acclimation. Okay, and finally, in the last, maybe 15 minutes or so we have, I want to talk about this other study. We're going to move to Wisconsin now. We're moving from the Southwest up to the Midwest. And we're going to think about aspen and ask the question, does intraspecific variation in NSC determine the defense response? Okay, so now I'm going to have you switch gears. We're going to talk about aspen. So aspen is a foundational species across, temperate North American forests. I'm sure you've seen it, although there's not a lot of trees around here, but if you go north, you'll see it. And so quaking aspen or Populus tremuloides can be fed upon by lots of different herbivores, including this invasive non-native spongy moth, formerly known as the gypsy moth, Lamantria dispar, right? And so this little critter here likes to eat on lots and lots of different tree species, right? But aspen is one of its preferred hosts. And what we see is that, it's an erupted species, so it will, their populations will increase. They will completely defoliate, right? We see extensive defoliation, and not just extensive, but repeated. They can come back year after year and defoliate trees. And it's really this repeated defoliation that stresses the trees out, right? It makes them susceptible to things like insects, drought and disease, and even mortality. Okay, so this is actually just from last month, August 2023, and Wisconsin sees worst spongy moth outbreak in over a decade. So it's kind of funny. We talk about it as like an invasive species, like, oh, this novel herbivore, but it's really been around for quite a while, especially in Wisconsin. So I don't know, I feel like it's going to become endemic pretty soon. But we saw a lot of spongy moth over the past decade. And really what I'm going to talk about is an outbreak that happened actually in 2021. Okay, so even though obviously there's lots and lots of aspen being defoliated here, they're not completely defenseless, right? So aspen has two classes of defensive compounds that we commonly think about and consider. So one are these phenolic glycosides, right? We put an X here because phenolic glycosides are highly effective against lots of different Lepidopteran species, including the spongy moth. So creating these, we would think that they would be highly defended, right? And so these are made from the phenylpropanoid pathway, as you can see here. But then there is another group of compounds also created from the phenylpropanoid pathway known as condensed tannins. I put a little X here, what I think is maybe bacterial, but it's supposed to represent a microbe, right? Because we typically think of condensed tannins as being antimicrobial. They're not effective against spongy moth. And so if you're creating lots of these, we would think you're highly defended. And if you're creating these, maybe not so much, at least in terms of spongy moth. Okay, and so now I want to bring back the non-structural carbohydrates, right, to remind you that these non-structural carbohydrates are providing the substrate and energy that is fueling the phenylpropanoid pathway, right? And so what you've seen here now is that there's an arrow going to both of them from here, suggesting that they share common substrates. And when we know that molecular formation and biosynthesis shares a common substrate, we can expect a trade-off, right? And so these are phenolic glycosides, normalized phenolic glycosides on the x-axis, and normalized condensed tannins on the y-axis. And these are all the genotypes actually at the garden I'm about to talk to you about. And what you can see is there's a clear trade-off in the production of phenolic glycosides and condensed tannins, where again, if you produce high PGs, right, you produce low condensed tannins. And so we think this is, our high defense trees against spongy moth. And then the opposite is true on this other extreme, where you produce high condensed tannins, but low to negligible phenolic glycosides. And so this is obviously, there's lots of intermediate chemotypes here, but we selected chemotypes at these extremes for this experiment. Okay, So in this study, what we did was we merged parallel lines of research. So there's lots of research suggesting that NSC dynamics, well, not lots actually, just a few, and more recently, that NSC dynamics are maybe genetically controlled, but there is more research on the defense side here suggesting that also constitutive defense dynamics are highly genetically controlled, right? And so what we want to know here is, how are they interacting with each other, right? If you know the genotype, but now you have an environmental stress, right? And are they still really, what is the role of genetics once you have this novel disturbance? Does everything fall apart, or can we actually, find some patterns and make predictions? So the question that we're asking here is, how does extensive defoliation influence aspen, NSC, and defense dynamics? And then kind of an add-on here is, how do the responses vary across chemotypic extremes? So really thinking about those highly defended trees making lots of PGs and the low defense trees that make basically none. Okay, so to answer this question, you need to find a genetic trial somewhere where you can grow these in the same place and they're mature trees, and someone's done like a decade of work, and you can leverage the chemistry and all of their data. And thank you, Rick Lindroth, and your awesome lab for that. And so we were lucky enough to be able to leverage all of that at the Wisconsin Aspen Common Garden, known as WisAsp. And if you say it really fast, it doesn't come out very well. So I had to really put that P on there. And so this is this incredible garden that we have near the University of Wisconsin at the Arlington Ag Research Station. And what Rick and his team did was actually collect over 500 different genotypes across a latitudinal gradient in Wisconsin, and then plant them in a block design with multiple replicates in this common garden back in, I think, about 2010. Okay, so what's great about this garden is that we can leverage those chemotypes, right? And then lucky for us, we actually, we went out in May of 2021 and we were like, oh my gosh, there's all these egg masses everywhere. And so we knew there was going to be a spongy moth outbreak, and we just threw the kitchen sink at this. And so, like I said, in 2021, this garden experienced a massive defoliation from spongy moth. This is LiDAR data showing red is full canopy and blue is basically bare ground. And so you can see major defoliation kind of happening here in early June. You see this wave kind of coming up from the southern part of the garden up to the northern part. It looks like some trees weren't defoliated, and that is true, but there's also a large gap here between the middle of June and early July where this does get bluer, right? And so we could use this event then to kind of ask questions about genotype, foliar traits, herbivory, the resulting defensive chemistry that is occurring here in the reflush, and what that means for future resiliency. Okay, so this is just kind of a little overview of all the work that went into this. We did so much sampling, both in the field and taking imagery with drones and using high resolution LiDAR and hyperspectral reflectance measurements. And so you can see all these drone flights. We measured foliar chemistry, leaf and twig NSCs, and then leaf, twig, plum, and root NSC collections. And we took them at key points, right? So we measured them pre-defoliation while the caterpillars were feeding. Then as soon as, we saw recovery, right, in kind of July, we call this early recovery, then late recovery in August, and then through senescence. So this is my postdoc cell. So this is literally neck-breaking work. It is terrible. He is a trooper. Okay, so what did we see? Basically, when we think about the defensive response, so remember, we have high defense trees and low defense trees. This is the relative change in phenolic glycosides. You can see during defoliation, no difference, but holy cannoli, during recovery in the reflush, we see the low defense trees just making tons of phenolic glycosides. So remember, they didn't really make any constitutively and are now rewiring, what they're doing with the phenylpropanoid pathway and making a ton of them, okay? Obviously, the high defense trees are also inducing, but not at the same level, right? And we see that in the early recovery period as well as in the late. And so what's interesting here is that by having this huge induced response in phenolic glycosides, these low defense trees, in essence, turn into high defense trees by the end of the season in terms of their chemistry. Okay, then when we think about condensed tannins, obviously for making PGs, it's going to come at a cost to those condensed tannins. And so you can see here in the reflush, for both chemotypes, we see a decrease in the production of condensed tannins relative to what they were just making constitutively, right? Okay. Oh, sad microbe. Okay. So that was with the defense. What about the NSC? So one question we kind of had was like, do these different chemotypes have different NSC kind of storage strategies? Can that help us, when we're thinking about predicting the defense response? And what you can see here, this is starch, and this is high defense and low defense. So we did see some differences. This is in the leaves. We saw that the high defense trees tend to store more starch in their leaves, about 4 times more, but only during pre-defoliation, right? During defoliation and during recovery, we actually see kind of a convergence in terms of leaf starch content. When we look at sucrose in the leaves, what's interesting is that there's no chemotypic differences earlier in the season, but wow, during the reflush, you see like all of the sucrose just kind of crash. And that makes a lot of sense, right? They're repairing a lot of things. They're making defense compounds. They're just, growing and things like that. So this decrease in sucrose makes a lot of sense. And I'm going to come back to this. Now, in the leaves, we saw that the high defense trees were storing a lot of starch in their leaves, but in the twigs, we actually see that it's the low defense trees storing starch, right? So they store it more in their twigs than in their leaves, and the high defense trees tend to store more in their leaves than in their twigs. But again, during senescence at the end of the season, we start to see convergence. So this is probably the most interesting figure that I've seen come out of this work, which is this relationship now between the chemistry and the NSC. So this is PGs, or phenolglycosides, regressed against sucrose. And as you can see, there's this negative exponential relationship between phenolic glycoside production and sucrose in both of the chemotypes. So this is very much suggesting some sort of threshold behavior, where when sucrose levels are high, right, we're at 8%, 6, 4, 2%, we see that phenol glycosides are really no greater than about maybe 5 or 6%, right? But once sucrose kind of drops below 1%, we start to see a huge increase in phenolic glycoside production, right, up here to up to 15% dry weight. And this is really happening mostly during the reflush, right? So when those sucrose levels are low, those trees are making tons and tons of phenolic glycosides, suggesting that maybe sucrose is serving as some sort of signal, but also likely being used as a substrate. Of course, if you have a negative relationship with phenolic glycosides and sucrose, we have a positive relationship with sucrose and condensed tannins because they're inversely related. Every line here is an individual tree. This is a repeated measures correlation analysis. And you can see that sucrose does tend to explain quite a bit of the variation in the condensed tannin data. Okay, so what is going on here? Basically, we have these high defense trees and low defense trees. Great. What I didn't show you was that actually there was no selective herbivory. We used the high resolution LiDAR to get LAI estimates, and we could estimate LAI pre-defoliation, during, and after, and they were no different. So spongy mouth does not select based on chemistry. But what we do see is that once these leaves reflush, these low defense trees that made basically no phenolic glycosides before now make a ton of phenolic glycosides. We see induced phenolic glycoside production in high defense trees, but not to the same level. And this increase is so large that actually the chemistry is almost identical. We also see that low sucrose concentrations may signal phenolic glycoside production and or serve as substrates. This threshold behavior is very, very cool. What the biggest take home here is that really in response to herbivory, NSC and defensive chemistry are converging, right? So pre-defoliation, clearly separate defenses, some differences in NSC storage. Once the spongy moth comes, stuff starts getting real, right? So we see low defense trees converging to look more like high defense trees, and we see NSC across the board kind of converging across in all organs. I didn't show you all the data. But this has led to more questions, right? So in the next season, we're like, oh, great, we're going to see all the spongy moth herbivory. Let's keep studying this. But actually, there was no spongy moth herbivory. It was only like 10%. They all died. And that was sad. But then we started noticing all of these cankers, and we noticed that all these trees started to become infested and infected with fungal pathogens. And so basically we have a defoliation event in 2021 that may be creating susceptibility to fungal pathogens in 2022. Our hypothesis is that by investing so much in phenolic glycosides and de-investing in condensed tannins, which are antimicrobial, it allows these endophytic pathogenic fungi to kind of just take root and infect these trees. We have no empirical evidence for this. This is just me spouting ideas, but we're looking at ecological legacies and ecological memory, and we have all the chemistry from 2022 to test this. So with that, I'm going to kind of leave you with this slide where I started talking about models, and I want to end here because our models, I'm not saying they're wrong, but I'm not saying they're wrong, right? And so they're not really doing a great job of predicting mortality. And this is because they tend to oversimplify what I'm showing to be very complicated and complex interactions between tree physiology, chemistry, and insects, right? And so what we're arguing is that we need to start including first principles of ecophysiology, secondary metabolites, and insect behavior into these models in order to, improve them. So the work we're doing is aimed at providing a more detailed understanding of physiological mechanisms by which tree defense and herbivores respond to environmental change. And the hope is that this will improve our predictive capacity, both in terms of mechanistic understanding, but also perhaps maybe with using remote sensing in the future to look at risks across landscapes. And so with that, I have a lot of people to thank, the Sevietta team, especially my co-PI, Henry Adams, Nate McDowell, Rick Lindroth, of course, for all the work that he's done at the WISAS, Common Garden, Phil Townsend, who's responsible for all the remote sensing work, and a lot of funding from the NSF and the DOE. So with that, I just want to thank you for your attention, and I'm happy to take any questions. Yes.
In the first part of your talk,
Which seems like forever ago. Yes.
so if I understood it correctly, you were saying the dead trees had higher levels of phenolics despite lower phenolics production.
They had higher levels of phenolics despite lower response to water potential. So in the end, when they died, they had higher levels in their leaves, but actually the rate of change to get there, right, was slower. Like they responded to their slope was lower than the slope for the legacy and the survival trees in terms of regressing that against water potential. So they weren't responding, they weren't making them as quickly, but in the end, they end up making more. So it could be that it's not, the end concentration that matters, but actually maybe how quickly you can actually respond to changes in water conditions.
Could they be getting them from other trees through underground root protection?
Oh, the phenolics?
Yeah.
I never say never. I don't think so, but these trees, their root systems aren't really, there's not a ton of grafting or things that go on like that. They do have ectomycorrhizal connections. There's lots of study by Kitty Gehring and her group that show that having those ectomycorrhizal connections actually do kind of confirm more drought resistance for lots of different reasons, but I think the molecules are too big to kind of make it that way, but they could definitely, people have suggested maybe substrate sharing and substrate acquisition that could maybe contribute. Yeah. So,
That's really good to think about insects and stuff like that. Drought, so I haven't focused on embolism. I was thinking, I don't know, keep harping on about phenolic stuff, but it was really cool. When I was looking at that data, it seems like could it also be explained by reduced flying flux and export out of the leaf, which happens with ABA.
Yep.
ABA levels accumulating leaves if you gird and branch. And so if you perhaps girdle branches of a legacy tree, you always increasing the knowledge, but you might also increase carbohydrates in the branch.
Yeah,
Attract users.
yeah. I mean, so it's funny you bring this up because these are things we're thinking about, and our funding runs out this year at the site. And we're just scrambling to maintain that legacy plot because yes, like how sustainable is this? Is this just going to keep going? Oh, well, that is, this is my crackpot idea that if it works out, we're all having steak dinner. But yeah, it's like, what is it? Like, there's clearly something about the chemistry of that tree where these bark, there's not a single sign of even, an attempt. Like it's like they can't see them or I don't know, or they just are deterred from them for whatever reason, but yeah, the volatiles, so we've been measuring volatiles at that site, monthly, basically, and so we have tons of VOC data. The preliminary data show that, once they reach a certain threshold, those volatiles just crash. At least for terpenes, but we're also measuring some other low molecular weight volatiles, like maybe acetaldehyde or maybe other, ethylene, like other volatiles that could maybe be playing a role. So even though the terpenes are kind of crashing, maybe that's allowing other volatiles to become more prominent as a deterrent. We don't know.
Can they sense sugar itself in the farm?
So I would imagine they can, because that seems to be the biggest reason for why they wouldn't choose them. There's just not a lot there for them to eat, and by the time the fungi can grow into like the ray parenchyma to extract nutrients or carbs or whatever, it's like they're so slow growing. So that takes a while. And so you really are relying on those carb reserves that are just there, at least in the immediate sense. And I think there must be something where they can sense sugar. We're always thinking about defenses, but the nutritive value is so important, both in terms of like free amino acids and carbs. Because like, where are they getting this from? Yeah, I mean, great question. We need to talk more. Well, we've already said this. Yeah, great question though. Yeah.
It'd be interesting to look at the landing when those things.
Yes.
Because there's a gustatory sense that goes on. It says, okay, this is the tree now I'm going to go in.
Oh my gosh. Yes. Well, how do we do that? I know. And that's the thing. It's like, well, we were just talking about maintaining like these remote field sites, and doing remote field work is so complicated because you can only go down for 1 week a month, and make all the measurements. And so, we already have taken so many, but that, I mean, I wonder if we could like do camera work. I don't know. Like, how can you, or just do a bioassay, maybe just force them to, to land and see. I don't know, but that's a great idea. Yeah. So that's really the piece that we're missing, is the actual, like, how do the insects, like, what are the behavioral responses to the physiology? And we've done some bioassay work with the different terpenes that we've measured, and we've done fungal growth plates with some of the terpenes, and it's, we see that actually with the terpenes in the phloem, we see that not all of them decrease with drought, but some of them do, and it's the ones that are the most effective against fungi that they vector. So we have some, we're getting there, but the insect behavior is the piece that's kind of the biggest black box. Yes.
In the aspens, after like they're defoliated by the moths, and then they put on a new flush of leaves, is there any chance you observe whether, like, when they're senescing then in the winter, is that senescence pushed off later to try and make up for
that's
lost photosynthesis?
yeah, that's such a good question. And so we have been monitoring that. So we've been flying, like, as soon as we start to see any activity there. We don't have those data, but it doesn't, it's complicated because the conditions year to year, and we get in the spring conditions, I think can really affect that. So there's like a bit of a confounding factor there. But we might be able to get to that, especially if we're seeing these maybe new disturbances. It would be interesting to see if that is being pushed as they're kind of getting hammered year after year. We have the data. I don't have an answer,
Okay.
but great. No, but we're thinking about it. It's a great question. Yeah. Yeah.
So when you were looking at the phenolic composition between the separate trees, were you able to determine if there was a difference in like the different specific phenolic chemicals between?
Yes. So what we're doing right now is using NMR. So we're using NMR to kind of look at not even just the phenolic diversity, but just the overall chemical diversity, which could be really useful, especially because we're just tapping into a very tiny bit of the remotely sensed data. You know what I mean? The phenolics make up a very small bit of all of the wavelengths and reflectance, information that we get. And so we're hoping to actually use the NMR and thinking about overall diversity and try and relate that back to some of the spectral data. But yes, I mean, with the phenolics, in opinion, we haven't done a ton of individual quantification. But it's something that I have a student who's interested in, but it's like, with the Aspen, it was easier. There was all this work that's been done already. Like, they're not easy to identify. But like I said, the NMR, at least, it might not give us individual compounds, but it will give us a broader sense of diversity. But so if what you're thinking is that it could be different relative, the concentrations could be different between the different types of phenolics, certainly. This was just a first pass. And would be, if it works out, would be an easy assay to do to link to remotely sensed data. Yes, or yeah.
I was curious if would enzyme, would have this hypothetical difference in vanilla composition, be due to enzyme kinematic differences, like there's an increased temperature because of the drought, there's also decreased water concentration.
Yeah.
Is that basically what I'm asking is, are trees ends with or I don't know.
I feel like we're starting to wax philosophical here. Basically what I mean, we can't get at the underlying mechanisms of the mechanisms with the types of measurements that we're doing. But I mean, it's certainly possible. Like, the data that we have showing or suggesting that these legacy trees don't respond as quickly, the trees that die don't respond as quickly, it suggests that there could be, just an enzymatic response. They're not maybe producing the enzymes as quickly, or for whatever reason, after a certain level of drought stress, if they haven't acclimated to that drought. You know what I mean? And there could be a lot of different explanations, but I don't really know. But the other thing about doing a field study like this is that we don't have any genetic information. So there could also be that component too that we've explored more with the aspen, but in conifers and especially in pinyon, we don't really have a good sense of how intraspecific variation in those types of dynamics really contribute to some of the patterns we see. But yeah, certainly, enzyme production or substrate or even the hydraulics. I mean, these trees have lots of diversity in terms of, just how they maintain osmotica. I mean, they're all isohydric, but there is a gradient within that isohydry that we see clearly.
Thank you.
Yeah. Yes,
So
John.
was the distribution of the pathogen presence in the work that's starting, is that specific to genotypes?
That's what we're doing right now. Yeah, because what we did was we're looking at it in terms of genotypes, but also my postdoc has gone through and he's doing NMR on all of the trees. And
you're still gonna get that flux.
Yeah.
So I don't, there's any microbial properties because
Yeah. Yeah. And so, and we have we've collected across the organs across the 2022 season and 23. So what? Oh, only in 2021. That would maybe be good though. Yeah. So, but it's something, I mean, it would be really interesting to see if the trees that in the next season maybe make more condensed tannins at the expense of the phenolic glycosides actually are less infected with the pathogens. And there's 16 different species and I mean, that is not something that's on my bucket list to explore, but just in general, definitely what we're thinking about. Yeah.
So what are your thoughts about patterns?
I have lots, so can it.
Here, we see this shorter drop period.
Mhm.
Illusions of rain versus places where you might have steadily drop periods and then a little bit of rain. Allocate these places.
I mean, gosh, that is such a great thought exercise that I've clearly had dreams about. And it's I don't know. I just know that it matters. Do you know what I mean? Like the pattern and the timing of drought stress really does matter. And it can be species specific. Like we are seeing, in some cases, legacy effects of drought, right? In some species, and then others, they can recover quicker, or maybe they don't acclimate as well and they die out. And it's like, so the timing, the duration, the severity, and that's the tricky thing about drought, is that we just don't have very good metrics to measure it, right? And so we use water potential, but that is like, maybe hold your ears, Scott, but so it's like the worst, if you're really thinking about drought as a cumulative metric, right? I mean, so that's the whole thing. It's not like something happens and then there's a response and that's forgotten. It's like there's kind of this legacy that's always built into trees, which is why they're so fascinating. But the water potential is just an instantaneous measurement of the tension at that tree at one time, and it doesn't really tell you about cumulative drought stress. Like, for example, some of these trees we measured and they were like -7 megapascals. I mean, just toast, right? And then they would get a rain event, right? So we measure it on a hot day, then a rain would come and we measure another branch and it was like -3. So is that really telling me, you know what I mean? Like, and so is that really telling me integratively how they're going to respond to a major disturbance? Probably not, but it's the best that we can do with this. But all I can say is, yes, timing matters, species matters, and yeah, severity for sure, but it's really hard to measure. I gave you no answer there, I'm sorry.
Maybe there is no answer.
Well, yeah, and I think it's just so complicated. That's the thing about drought, it is just so complicated. I mean, even within the site that we measure, you can see there are like there's pinions up on mesas, there's pinions down in arroyos, so there's so much variation just in and of itself. But I will say in this species, that root architecture is so key to survival. Being able to tap into water, deep water is really important.
All right.
Gosh, thanks everyone.
Yeah.
Stop share. Do you want me to turn this off? Should I just disconnect this or?
Go ahead and just unplug.
Okay.